Hello, everyone, and welcome to the Cell and Gene Therapy Manufacturing Digital Week, brought to you by the producers of the face-to-face cell and gene therapy manufacturing events, visiting Boston in September, Amsterdam in December, and Kyoto in February 2021. My name is Brianna Costafret, and I'll be your host for today's session titled Improved T-Cell Function Through Metabolic Modulation Towards an Early Memory Status, sponsored by Thermo Fisher Scientific. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, submit your questions into the Q&A window on the left-hand side of your screen. In 24 hours, you'll receive a link to watch the recording of today's session. And also, please note the resource list box on the right side of your screen to download an additional resource. Now let's begin by introducing our speaker from Thermo Fisher Scientific. Today we have Evan Zinda. He's in charge of research and development for bioproduction at Thermo Fisher Scientific. Thank you very much for joining us today, Evan. I'll turn over to you to begin your presentation.
All right, thank you very much. So thank you all for tuning in this morning. My name is Evan Zinda, as she said, and my expertise lies in T-cell therapy process development, which we view as an interrelationship between the products and the processes. And today I'm going to tell you about how we can actually tailor the products and processes towards improving the workflow and potentially improving the therapeutic performance. So this chart shows the projected market increase for cell therapy over the next 7 years. And you can see that last year we were a little under $500 million, and this is expected to grow an annual compound annual growth rate of 34.5% over these next 7 years, meaning we're still at the very beginning of this, and it's going to boom. But all of these projections rely on the assumption that we will be able to meet many expectations and requirements and also be able to overcome many of the challenges that we face. Some of these requirements are listed here. So these are a lot of the staples that allow us to basically create a perfect, or as good as we can, create a very good cell therapy. And they mainly fall under a few buckets, such as consistency. And you can see consistency in performance and control, as well as manufacturing here. Safety in regulations, as well as clinical trials, so that this is safe for the patients, and also availability. So by that, we're looking at the guarantee in supply chain, as well as being able to scale up and scale out these processes so that we can get them out to more patients. We have already had many achievements in this field, and those are kind of summarized by these two bullets on the left in Kymriah and Yescarta, the therapies that have already been approved and have proven to be effective in patients. But there are a whole slew of challenges we still face, such as those listed on the right. And these fall into several categories, some of the same ones that we spoke about on the previous slide, such as the safety, consistency, and availability. But in addition to that, there is also new technologies needed, especially when it comes to approaching solid tumors and allogeneic workflows. So with regard to the solid tumors, they pose additional challenges due to the fact that these microenvironments don't usually allow these cell therapies to readily go in there and do their jobs. And these tumors are also, they have their characteristic heterogeneity doesn't allow for us to target antigens very easily. And then looking at the allogeneic challenges, that's mainly due to upfront costs and safety. And then moving on towards those that I already mentioned on the previous slide, some challenges that come in the face of safety and consistency include completely being able to close the system and automate the system. So if we are able to have a completely closed and automated system, we will be able to avoid human error and potentially make these much safer and have much less contamination to the patients. In addition, there is also process characterization. And what I mean by this is having a process and a workflow that is completely populated with consumables and devices that are dedicated to cell therapy and were developed with cell therapy in mind. and not repurposed from some other intention. And this will make the processes more efficient and presumably more effective. And then finally, maybe the largest challenge is the cost. And this includes the cost of goods and services, as well as the cost to the patients. So this can actually be reduced if we were able to completely close and automate the system. We would have less of a need for labor, utilities, cleaning supplies, and PPE. But there still needs to be a big reduction, as these therapies tend to be almost $400,000 to $500,000 each. So the key to creating a sustainable market is to reduce the cost of the drug, and we think a lot of that comes from the manufacturing process. So this is a schematic of the entire workflow process, and we can see that there's 3 buckets containing about 15 individual processes. And for the purpose of today, I'm going to be focused on the manufacturing workflow, mainly from cell isolation to cell harvest, wash, and volume reduction. And we believe that this is a very crucial point in the workflow. And so next I want to show you how some of our customers and we tend to measure the manufacturing success based on the cellular outcome. So here I've listed 6 bullets that kind of encompass a lot of the parameters that can let you know whether or not you've had a successful manufacturing process based on the cells that you produce. So starting with T-cell proliferation, obviously we need the cells to grow in order to get a dose that is large enough to cure the cancer that we're targeting or the other disease that we are targeting. But in addition to quantity, we also need quality, and these other 5 bullets kind of fall under that category. So starting with the viability, this is kind of just a measure of the cell health and happiness, and we want to see high viability throughout the workflow. The CD8 to CD4 ratio is a bit of a gray area. So some companies and some groups tend to want to see a very good balance between the CD8 and CD4 numbers, while others prefer to have more CD4 or CD8 cells. The important message is that both of these cell populations are very important to the immune response. It doesn't really matter how you get there. As long as you make something effective, there are different ways to get there. And then the lower right is the engineering efficiency. So we just want to make sure that we have efficient knock-in and knock-out of the genes of interest to ensure that we have as high of safety and efficacy as possible. These lowest 2 bullets have actually gained the most attention recently, and that is the phenotype and functionality of these cells. So we'll get into the phenotype a lot throughout the next few slides. And what I mean when I talk about the phenotype is usually people are looking at the activation status, differentiation, and exhaustion. And with regard to the functionality, people often use cytokine analysis, which is what I'm showing here, as well as killing capacity and engraftment and persistence within the host. And I'll show some of that data later. As I said, I wanted to start off with the importance of the phenotype. So this has been kind of a moving target over the past few decades. If you would have looked at the literature 20 to 25 years ago, you would have seen papers stating that effector T cells made for the best cell therapies. However, there has been a shift, and in the past 10 to 15 years, you tend to see almost a complete consensus that the central memory, stem cell memory, the younger, more immature cells actually make for the best phenotype for cell therapy. And this quote on the left actually kind of supports this. And this is a quote out of a Rosenberg paper almost 10 years ago, and it states that the central memory, stem cell memory cells actually show higher tumor clearance and engraftment and persistence. So that's kind of the aim. We want to fall into that left side of the spectrum where these cells show high proliferative potential, high survival, and are very responsive and productive with regard to the immunostimulatory cytokines. As they shift over to the right, they lose all of these qualities. They become more cytotoxic, but they aren't long-lived, and they don't survive very long, and they don't really orchestrate immune responses. But a major problem is that we don't control the input to the workflow, and that is the patient cells. And oftentimes they present with pretty glaring flaws, the main two being illustrated here, and that's donor variability as well as low cell numbers. So you can see over on the left that this is a graph showing the CD8 positive T cells within patients versus healthy donors. And those cells are actually seen at a much lower rate in the patients. And this could be potentially detrimental, as these cells are very important to the immune response. And then moving over to the middle and right side panels, you can see these are more along the lines of the donor variability. So starting in the middle, here we have stained for two central memory markers, CD62L and CCR7, which will be kind of a common thing throughout the rest of this talk. But staying on the left side of it, you can see that the double positive staining cells, there is a very wide range of expression of these central memory cells, ranging from about 12% to 73% in this donor cohort. So we could assume that the patient showing about 12% central memory cells would probably not fare as well as the patient showing about 73% central memory cells if these cells actually maintained this phenotype throughout the workflow and after manufacturing. And then moving on to the right, it's a similar story with the CD8 to CD4 ratio, where we see a very wide range of the CD8 to CD4 ratios for these donors. And the assumption is that these cells also would not behave the same when turned into a cell therapy, and the patients would not have the same outcome. Our goal is to be able to take any patient cells and manipulate them and engineer them and grow them into safe and efficacious therapies. So we want to erase those variabilities and create a very, very safe and effective therapy no matter what the starting point. And we're not the first people to come up with this idea. This has been around for a few years now, and one of the first groups we saw doing this was actually Carl June's lab over at UPenn. And they actually looked at patients from a CLL clinical trial where there was 26% durable response, and 74% of these patients didn't show that durable response, and they called these the non-responders. And we'll do the same throughout here. So the idea for them was to look at expression profiling, comparing the responders and the non-responders. And then they wanted to be able to take advantage of these differences, some of the glaring pathways that were turned on and turned off, in an attempt to try to move the non-responders over towards the responder side. And we believe we could do the same kind of thing, but using the manufacturing workflow. So as I stated up front, I think, we believe the manufacturing workflow is a combination of the products and the process. And if you have best-in-class products, and this includes the devices and the consumables, and you combine them with very, very thoughtful, both from the standpoint of biology and simply being able to close and automate these systems, you have to have very thoughtful processes that consider every single effect on these cells. If you combine those products with these processes, you can get the desired performance. So looking again at a different schematic of this workflow, we see that the whole thing takes about a little over 2 weeks. In addition, we see that the activation expansion phase is about 2/3 of this whole process. So we thought it was logical that we could attack this segment of the workflow in trying to create products and processes that could drive these cells over to a favorable phenotype. And the first logical place we went to was the tissue culture media. So when I talk about culture medium, what I'm referring to is the basal medium, the medium supplementation, and the cell stimulants, such as interleukins and growth factors that get put into the media. And we thought this was a very logical place to start because the media is in direct contact with the cells for that entire 10-day span throughout the activation expansion phase. In addition, it's relatively easy to manipulate from a biological standpoint. We can easily, formulate with new small molecules in there in order to turn on and turn off pathways. And then over on the right, this data isn't meant for anything that's going to be a take home from the whole talk. It's just meant to show you that the media can have pretty significant effects on both the growth and the phenotype that are resultant from the expansion. The workhorse that we tend to stick with is the CTS Optimizer serum-free and phenol red-free expansion medium. So this medium actually represents a pretty great achievement for us in that it is one of the media used to produce one of the clinically approved and proven to be efficacious therapies that I spoke about before. So we already know that this media can be part of a workflow that produces efficacious cell therapies, and that's a pretty big accomplishment. In addition, it's often supplemented with CTS immune cell serum replacement. And these can actually form, and both of them they do overcome challenges, some of the challenges that I mentioned earlier. And these include that because it is serum-free, it is safe and consistent, and the supply chain is guaranteed. And because it is phenol red-free, it is very friendly to automation. Sometimes the pigment can actually interfere with automation sensors, so not having phenol red is a benefit as well. And this media can lend itself to basically any closed and automated system in a safe and consistent way. And that's a major, major benefit. So now if we start to look a little bit more closely at the performance, these data show some of the flexibility of this media in both rocking and static bioreactors. So you can see over on the left that we get robust growth in both static and rocking bioreactors, and that there is an improvement in the growth using the rocking motion, as well as even a little bit further using perfusion. Over in the middle, you can see that this media supports high viability in all three of these bioreactors, and this is actually slightly improved with automated control. And then on the right, we're looking at central memory, stem cell memory markers, CD27 and CD62L, and we can see that this media supports a very nice percentage of cells displaying the central memory desired phenotype, and a low number of cells displaying the double negative effector-like phenotype. And also well documented is that Optimizer with ICSR can actually perform just as well as serum-containing media. So here we see data showing growth, viability, and phenotype comparing Optimizer to serum-containing media, and we see that the growth, viability, and CD8 to CD4 ratios are almost identical. And there is actually a slight benefit to using Optimizer with ICSR when it comes to the central memory expression. And I should say that all of this data was the average of 3 donors and is representative of many, many more donors than that. But over in that lower right differentiation chart, you can see that there is a slight increase in the double positive staining and a decrease in the double negative staining. So another, good quality of this media. Less well known is the in vivo work that we did in collaboration with UPenn. And here we grew CAR19 cells in Optimizer with ICSR or serum-containing media, and then we inoculated these cells into mice bearing B-cell tumors that express luciferase. And by day 13, you can see that there was very good tumor clearance using the cells grown in both of these media. However, by day 49, we can see that the tumors had come back when the mice were treated with the cells grown in serum-containing media. However, those grown in Optimizer with ICSR still showed very nice tumor control in 4 out of 6 mice. And these data could actually be validated by the engraftment and persistence. So over on the right, if you look at the day 14 circulation of cells grown in these different media, you can see that there was basically a similar amount, or there was at least plenty of circulating cells grown in both the Optimizer and in the serum-containing media, and that corresponds to the day 13 results. However, moving out to day 40, corresponding more closely to the day 49 results, we see that almost all of the cells that were grown in serum-containing media have been cleared. However, those cells that were grown in optimizer with ICSR still persist, and this is likely the reason that we see a durable response. And this is all great data, and it does prove that the media does make a difference in the cell therapy efficacy and performance and even the safety. But it doesn't really help us at the moment. We've kind of run into a wall as a field, and we haven't been able to successfully launch any therapies that target indications other than the B-cell indications, such as those approved therapies that I mentioned earlier. So we need to do a little better. And previously, almost all data or almost all media has been developed with growth and viability as the primary endpoint that is considered. And then phenotype is kind of just a bonus or a passive aim. Here, we want to actually develop a media that will drive the phenotype. So for this purpose, we're going to try to use our optimizer media. We already know that it does work well and it is capable of producing efficacious therapies. We just want to improve it a little bit more to try to fine-tune those cells that come out of expansion. And so the idea here is to use the media to drive cells from the right side of the spectrum over to the left side of the spectrum towards that central memory phenotype. And to do this, we had to exploit a difference within these two populations. And that difference was actually their preferred methods of metabolism. So you can see that central memory stem cell memory cells actually employ oxidative phosphorylation to produce energy, while those that shift down towards the effector side employ the less efficient glycolysis. So our thought was that we could inhibit glycolysis and push these cells from the right over to the left side of the spectrum, away from effector towards the central memory cells, the ones that are associated with a higher increased efficacy. And so by doing this, we would create a better therapy that may be able to convert some of the non-responders over to responders. So we created an optimizer with this exact intention, a next generation prototype of optimizer, and then we have some preliminary results showing the results in a traditional expansion as well as a longer term expansion. And here I'm showing growth and viability, and the green is the next generation prototype and the black is the parent optimizer. And you can see out to day 10, there is basically no difference in growth. And this is a traditional, kind of like an autologous workflow. And then as you move from day 10 out to day 15 and even further, you see that there is a gradual increase in the growth with regard to the prototype media compared to the parent media. So the take home here is that the growth is at least as good. Assuming we get the same amount of growth, but we increase the number of central memory cells, we would then kind of extrapolate that we should have better efficacy if all of our hypotheses hold true. And then looking over at the right at the viability, we see no difference in viability throughout this entire workflow. So now let's look at the primary aim, which was to investigate whether this media could actually drive a younger, more immature phenotype. So here we stained for a couple different panels that indicate central memory and stem cell memory populations. And again, the green is the prototype media and the black is the parent media. And we actually normalized the percent of central memory cells in our next generation media to the number from the parent optimizer in order to be able to clearly see how big of an increase we got. And you can see that in both these panels, we see about a 15% increase in the number of central memory cells for the populations grown in the prototype media compared to the parent media. And this is very significant because it's speculated and there's models that predict that every one of these cells can be responsible for the death of 10,000 tumor cells. So every little bit helps, and this is a pretty significant jump when you think about it that way. In addition to memory, we also looked at the exhaustion. And here you can see that we stained for PD1 and TIM3, two markers for cell exhaustion, and we saw that there was significant reductions in both of 10% and 46% respectively. I should state that there wasn't a high amount of exhaustion using the parent optimizer in the first place, but this was just interesting data to us nonetheless, and it is supportive of a more favorable phenotype. So this was a great example of how a consumable could actually improve the cell therapy workflow. I should mention that we do still need to correlate these improvements in phenotype over towards improvements in efficacy, but this is fairly preliminary, and we're still seeking out some collaborations and partnerships in order to test some of this stuff in a clinical setting. But moving on, next I want to get into how we can actually improve the workflow through more thoughtful design of our devices and equipment and hardware that is used. And here we can see again this highlighted region where it's the manufacturing workflow that I told you I would be sticking to. And there are many areas within this workflow that require equipment or could use equipment anyway, and those are the cell isolation, electroporation, expansion, wash and volume reduction, and a controller to integrate all of these and keep it closed and automated. When thinking about this, we instantly gravitated towards the cell isolation step as a step that we were pretty unsatisfied with what was on the market from the standpoint of being able to easily automate and keep closed, the entire workflow, as well as just being friendly to any kind of integration. For this purpose, we looked into developing a counterflow centrifugation device that would be able to help us isolate the PBMCs and T cells from the original population. And by doing a counterflow centrifugation, we'd be able to knock off some other tasks within the cell therapy workflow as well, such as the wash steps and the concentration and harvest at the end of the workflow, more downstream. So counterflow centrifugation offers several benefits to this workflow. First, the cells are loaded into the chamber, and then you can actually separate out your cells of interest. In this case, it would be the PBMCs and T cells. You can separate those out by basically manipulating the centrifugation speed as well as the fluid flow rate out of the chamber. And so you can separate the cells out by density and size and get your population of interest. But beyond that, you can actually do some downstream steps as well. And like I said, one of those is the wash step. So here you can, again, load the cells in the chamber, and then you can actually wash out the buffer they were in and put them in a new buffer and then harvest them out. And this could be useful from the activation to gene editing steps, and also from taking them out of gene editing and putting them back into expansion. And then finally, this machine can be used for the harvest and wash and volume reduction at the very downstream portion. So here the cells will just be loaded into the chamber, and you can basically concentrate them and harvest them into as low a volume as the instrument will allow. For this purpose, we developed the Rotea, and this was developed specifically with cell therapy in mind. So it offers many benefits, such as it has pre-programmed processes for all of those steps that I just spoke about, and you can use these as templates and manipulate them any way you want to fit into your workflow or your intentions. But getting into some of its attributes, it can obviously do the separation, washing, and concentration, like I said. It also lends itself very well to basically integrating into any closed and automated workflow, and it can be scaled up fairly seamlessly as well. Getting into some of the performance attributes, first we wanted to look at its ability to recover viable cells, recover and maintain cell viability. So on the left, we put 5 billion cells through the process of recovery, and we see that we were able to recover about 95% with a viability of about 95%. And this was a very high cell number, so these results were extremely encouraging. And 5 billion cells is the max we can load into our chamber. And then going toward the middle, here we wanted to see how low we could reduce the recovery volume. And we took 500 million cells and we recovered them in 5 mLs, 10 mLs, and 20 mLs. And you can see that the recovery statistics were basically the same between 5 ml and 20 ml. You can see that we recovered about 100% of the cells and at a viability of about 95%. So this was encouraging, and then we wanted to see and verify that we didn't damage the cells in any way. So here we're looking at the CD4 and CD8 numbers pre and post processing. And you can see the before is in blue and the orange is after the processing, and there is almost no difference in the CD4 and CD8 populations. And we already stated that we recovered almost every single cell. So this is very encouraging, and it's evidence that we aren't damaging the receptors on these cell surfaces. And in addition to this, I should also say that we did similar experiments regarding the differentiation markers, and we saw the same results. We weren't damaging the receptors on the surfaces at all. So now let's get into comparing the Rotia isolation to a more traditional phycol separation, which is manual isolation. And on the left is phycol and on the right is Rotia, but I want to start in the center where I have them side by side, and you can see that the viability was identical just about between the phycol separation and the Rotia isolation. However, there was a higher cell recovery with the Rotia isolation. And then if you move down to the orange circles, you can see that within the cells recovered, there was about 96% to 97% leukocytes in both of these pieces of equipment, and that the T cells within those leukocytes represented about 80% of the population. So if we're recovering the same amount of T cells by percentage in the population, but we're recovering more cells, we're actually recovering more T cells. So that's pretty encouraging to us, and it basically means that we could potentially reduce the duration of the expansion before we have to basically put a adequate dose into the patient, and this could reduce the cost of the manufacturing overall. Furthermore, another fairly large benefit that I didn't mention is that phycol separation tends to take about 2 hours, while the Rotia isolation took less than 30 minutes. The programs that we placed into the machine are actually about 20 to 25 minutes. So this is a very significant difference as well. This is cutting that processing down by 75%. So this could also reduce the costs just simply by time, let alone the technician necessary. So these are all great benefits. Let's just go over all the take-homes on this piece of equipment. So as I stated up front, you can have a lot of tasks accomplished by this between the cell separation, the wash, and the harvest and concentration at the end. In addition, it fits into most any workflow, and it can be easily closed and automated and scaled up. And then from the performance standpoint, you actually see that the cell recovery is better than traditional phycol separation methods, and it is way faster as well. So this was an example of how we can use equipment to improve the workflow, mainly from a safety and cost standpoint. Now I want to get into how we can actually tailor processes to whatever our intentions are. And so we've actually investigated almost every nuance in the workflow and tried to just see how we can change the endpoint with regard to the cells. And here I'm listing several of these processes and or conditions that can be altered in order to affect the cellular product. Starting with up top, the environmental conditions such as gassing and pH. I'm specifically going to get into the cell density and length of expansion, And then others include the vessels. We already saw that different vessels can result in different growth. And what I mean by scheduling of interventions is when and how you're doing the gene editing, when you do feeds and how often you do feeds and stimulation, as well as even bead removal. And then, also looking at the bottom there, the different methods for activation, gene editing, and cryopreservation are also well known to have an effect on the cell outcome. So, as I said, I wanted to start off with the cell density. Traditionally, most groups and most people tend to expand their cells at a density of 500,000 cells per ml. And this doesn't include the activation and gene editing steps, which often need slightly higher densities for those 24 hours or whatever. But for the rest of the expansion, people try to maintain 500,000 cells per ml has been usually the rule of thumb. But we wanted to look at lowering and increasing this number and how it would affect the cells that came out of the process. So starting with growth and viability, we can see a fairly linear relationship. And the lower density, here I'm showing you 0.25 million to 0.75 million. The lower density, 250,000 cells per ml, actually has much, much higher growth than 500,000 and 750,000. And I should state that this is, again, for the average of 3 different donors, but it's representative of many, many donors. And then moving on to viability, we again see a linear relationship, a slight increase in viability as you lower the density that the cells were maintained throughout expansion. Moving on to the phenotype, on the left, you can see, again, like a dose-dependent curve almost, where the lower the cell density, you get a higher CD8 to CD4 ratio. And this could be very important. As I mentioned earlier, patients often present with low CD8 numbers, so this would be a potential method by which we could maybe erase that variability. And then moving on to the differentiation on the lower right, here we are staining for CCR7 and CD62L, and you can see that at the lower density, there is an increase in the preferred phenotype, the double positive phenotype, and there is actually a decrease in the double negative or effector phenotype. So these results they're pretty cool to us, and they are just an example of how something really seemingly mundane and simple can have fairly large effects on the cellular outcome of your workflow. So here, now I'm talking about the length of expansion and how that can affect the cellular product. On the left is growth. So obviously, if I grow the cells out to day 19, I would assume there would be more cells than there were at day 10. And I should say that this data comes from a rocking bioreactor with perfusion. And you can see at day 10, there's about 120-fold expansion. And then at day 19, this is increased up to about 50,000-fold expansion. That's fairly obvious that you would get more growth the longer you left them in culture. But it comes at the cost of phenotype. So if you look over on the right, the purple bars correspond to the day 10 phenotype, and the green bars correspond to the day 19 phenotype. And you can see that you lost more than half of the central memory population between day 10 and day 19, and the effector population was increased by about a factor of 4. So you got a lot of growth, but now you don't have cells that perform as well. You might not actually make any difference in efficacy this way. It definitely wouldn't improve it. But we got to thinking, we had that prototype media which can actually drive cells to maintain a central memory phenotype in traditional workflows out to day 10. What about out to day 19 where we push these cells a little harder? Could this media still maintain the younger, more desirable phenotype? So here we show these results, and again, we're comparing the parent optimizer to the next generation prototype, and the prototype is in green. And we normalized the prototype numbers to the parent optimizer numbers, and we can see that there's a 2 to 2.5-fold increase in central memory and stem cell populations, stem cell memory populations as indicated by CCR7, CD62L, and CD27 staining. So this was extremely impressive to us. We really pushed these cells to the limit, and to a place where these cells are normally driven to age and just become way less effective. But the media actually does do a good job of maintaining these cells in their younger, more vibrant phenotype. So that was an example of how we can use the process to improve the workflow outcome. And that's all I got for you today. I'm just going to finish up with a couple of messages that I wanted to emphasize before I get out of here. And the main overarching theme here was that products plus process equals performance. So if you have best-in-class products that are very well designed and dedicated towards cell therapy, and you combine them with biologically thoughtful processes, you can achieve whatever your desired outcome is. So basically, for us, that is that we want to be able to erase any variabilities between the donors. And for today's talk, we wanted to be able to create a more central memory-based population, no matter what the donor sample looked like. So in the near-term future, some of our goals are, we obviously want to keep characterizing this next-generation media and validate that the phenotype does correspond to improved efficacy. And as I mentioned, we are currently trying to enroll some more collaborations and partnerships to gain access to a clinical setting. And beyond this, we also want to take this a step further and employ our high-end multi-omics capabilities. For this purpose, we would take responder and non-responder samples, and we would subject them to omics analysis and try to pinpoint some glaring differences that we could maybe try to exploit in media development, and add in some ingredients into the media that would drive certain pathways to be on or off and create more of a homogeneous population and convert some of these non-responders to responders. And then finally, we are also developing more devices to aid in creating closed and automated and friendly and effective workflows. With that, I'd like to thank you again for tuning in, and I've included my email here in case you have any inquiries or questions after you leave. Everyone be safe, and I think I'll take a few questions now.
Thank you, Evan, for a great presentation. We've received a few questions already, but we'll give the rest of you a moment to enter your questions in the Q&A box to the left of the slides. Before we begin the Q&A, I'll run through some brief announcements. First, I'd like to thank Thermo Fisher Scientific for sponsoring today's event. Next, I'd like to quickly draw your attention to our face-to-face cell and gene therapy manufacturing events, visiting Boston in September, Amsterdam in December, and Kyoto in February 2021. And also make sure you still check out that resources list on the right-hand side of your screen. Now let's go back to Evan for the Q&A. All right, Evan, here's our first question. When you talk about 20 to 25 minutes for processing in the Rotea, to what starting volume is that referring?
1 liter, and that's using a moderate flow speed of 50 mLs per minute, which is the default speed in the programs. And that speed is meant to achieve extremely high recovery and high purity. So you can actually increase that speed or decrease it depending on your aims. If you want a very, very fast process, you can increase that speed, but you might recover a little bit less. But for that purpose, you can do 1 liter in about 20 to 25 minutes.
All right, thank you, Evan. When will the Rotea and next generation media be commercially available?
The next generation optimizer was projected to be launched in Q3 2020, but I assume that might be slightly delayed now. That said, we are still taking on alpha testers, and if you would like to be part of a collaboration or are interested in doing this, just shoot me an email and we can figure that out. And with regard to the Rotea, there has been limited release in the first half of this year Again, that's being slowed a little bit right now, but if you would like more information about that or scheduling a demo, again, just shoot me an email and I'll put you in touch with the appropriate parties.
Let's see here. Next question in our queue. Was the data for the prototype media created using healthy or unhealthy donors?
The data that I showed was with healthy donors, but since making these slides, we've actually done some more work in unhealthy donors, and it's extremely preliminary, and I think it's only with 4 unhealthy donors at the moment, but we do see very similar results, and in some cases, we actually see a more pronounced benefit to growth and phenotype. And this could possibly be due to the fact that these donors are often a little bit older, so their cells, may have more of an old phenotype, that like an effector phenotype, so we have more room for improvement. But again, more work needs to be done to draw any conclusions here, but we do see the same effects in unhealthy and healthy donors. Done.
Okay, wonderful. Thank you, Evan. All right, here, let's see. Our next question is asking to go back on slide 38, so let's go do that now. And here we go. Can you describe the methods used for the perfusion experiment on slide 38? Let's see, couple part question here. How did you boost the fold expansion so high, and what did the perfusion schedule look like? Did viability dip?
Okay, yeah, that's a bunch. It was done with Optimizer in the Zuri bioreactor, that whole thing. And we used automated control of gassing and pH, which has a benefit usually. And then I'd specifically attribute the high growth to using low cell density during expansion, as I explained during the talk, until we reach the volume capacity. And then once we reach the volume capacity, we start perfusion, usually about 1/2 volume per day, once the cells reach 1.5 to 2 million cells per ml. And then we increase this rate gradually up to 2 volumes per day when the density reaches 20 million cells per ml. So basically the idea is just to keep trying to supply the cells with unlimited gas and nutrients throughout, which I think is the reason that the low density actually has such a benefit on these cells. The viability was extremely high throughout the whole thing, right up until maybe the last day or two, but it never dipped below 90, even at that point.
All right, wonderful. Okay, Evan, we have time for one last question, and here we go. What is the lowest volume in which the Rotea can harvest cells?
So the lowest volume that we recommend is the 5 mLs that I presented. We've gone down to as low as 2 to 3 mLs, and it is possible to recover in there, but you will sacrifice some consistency. When you get into that low of a volume with a lot of cells, that you can have, just little areas where a droplet or something might collect, and it makes a significant impact on your recovery. So 5 mLs is very efficient and effective. Going below that is a little bit more inconsistent, but it can be done between 2 and 3 mLs.
And that's all the time we have for questions today. Thank you very much for a great session. If anyone submitted a question that wasn't addressed, keep in mind that the speaker will reach out to you directly. This session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. And before you log off, please take a moment to complete the feedback form so we can continue to improve your Digital Week experience. On behalf of Connect 365 Life Sciences, have a great